Oh, this is a very interesting slide, look at this. We have said that at the level of the Bowman's capsule, when the ultrafiltration is happening, okay, because of the charge on the basement membrane and because of the size of the protein molecules, they resist filtration, but no system is perfect. So, in case some protein molecules are filtered, okay, the nature has come up with a beautiful solution. What is the solution? The solution is here. I am looking at a cell. What cell is this? This is a cell from the proximal convoluted tubule. What face I am seeing? I am seeing the apical side. Can I see the villi there? On the villi there, the author has given a tiny brownish patch there. Can you see that patch there? That is a receptor which can identify proteins. It can not only identify proteins, it can bind with the proteins. So, any proteins which by mistake or whatever I do not know, when they have been filtered, they will bind to this molecule which I can call as a receptor, okay, and it binds, and then so this green it codifies the protein molecule, it binds to the protein molecule, and once it binds, it will be taken in by the process of pinocytosis. We have done that in school, pinocytosis, and then it will pinch off, it will go in the cytosol. There may be another lysosome, okay, another it will fuse with a lysosome, okay, the lysosome may release certain H ions, the protein will be digested, it will be broken down into amino acids, and those amino acids will finally be released. Nature does not want to lose any protein molecule. So, by mistake, if any protein molecule has gone into if you do not save it, it will go, okay. So, this is a mechanism, and you better do it at the level of proximal convoluted at the beginning itself. Okay, let us get rid of all the, not get rid of, I am sorry, let us make sure that protein molecules that we are afraid of losing, okay, we pick them up, and since the molecules are so large, we are talking about protein molecules, they are so large, okay, it is just not possible to provide a carrier, and no, we need a bigger system, and that bigger system is nothing but this pinocytosis by which we can, this is another very, very interesting phenomena. I will request you to focus on this. This phenomena is called as transport maximum. Now, listen to this. SGLT2 is there. Hello? SGLT2 is there in the apical or basal? Apical. Okay, and it plays a key role in picking up the glucose molecules. Okay. Now, imagine what would happen if the number of SGLT2, am I right? SGLT2 protein molecules is limited, not infinite, not limited, there is a limit to that, okay. Limit to that means what? As long as certain number of glucose molecules are coming, okay, they will take, but if you give more and more and more, you will reach a point of saturation, okay, and if you reach that point of saturation, after that what will happen to glucose in the filtrate? You will pass it in urine. You will pass it in urine. So, now I will simply ask you a question that here is a healthy human being, whose blood sugar is controlled within the range of 100, 120, 130, 40, whatever milligram, okay, and you take the urine sample and find that it is totally devoid of glucose, But there is another person who is a patient of diabetes and he has blood sugar level of say 200, 250, 300, then you start getting the traces of glucose in the urine, and there may be more than traces, you can have higher concentration of glucose. Can you relate this to my earlier observation? What is happening? Saturation. There aren't enough number of SGLT molecules to make sure that all the okay, and this phenomena is called as transport maximum. Makes logic? Sounds, yeah. Transport of what? In this particular case, there are others also. Transport maximum. What do you call it? This phenomena is called as what? Transport maximum for substances that are actively reabsorbed. So let us study the phenomena of what you call as transport maximum. We are still in proximal convoluted tube, we have not gone ahead, huh? We are still in proximal convoluted tube and we are discussing the physiology of the glucose absorption, and there is a constraint on that, and that constraint we call as transport maximum. Now, it has been found that plasma glucose concentration on the x axis. And as a normal person, you are if you are fasting, you are about 80, you are here, and it is post meal, you are about 120, 130, you are here. So, normal more or less, and that is filtered, glucose filtered load is about also so much. So, whatever is being filtered is being absorbed, is being what? Is being absorbed. But once you cross the plasma, glucose in the plasma to beyond 200 or 250 or so, then reabsorption is not possible. And then you start what? You start excreting glucose in the urine. It starts appearing. Okay, I mean, this graph tells us actually the values at which you may start, and this is okay. So sweet glucose. Okay, you have reached a point of saturation. It may be about 300, which is a lot. We have a lot of margin. Okay, in a healthy person, the blood glucose level never goes above 120, 140. Okay, but you are still okay. I mean, even if it goes to 200, 250, okay, your SGLT is still enough to make sure that the glucose molecules are being absorbed. But the moment you go above 300, okay, then it starts. You have reached your transport maximum, okay, and then the glucose will start appearing into what? Into urine. Are you getting the concept slowly? We are just considering the point of saturation of the process of reabsorption. Can be done, but it crosses a certain limit, it can't be done. Now, this process of this is a classic example of glucose, but that's also true for a lot of other substances which are being reabsorbed. Glucose transport maximum is 375 milligram per minute. Okay, are you with me? So much can be, this is the limit, okay, maximum limit this. Okay, then for phosphate also is actively absorbed. It's about 10 millimoles per minute. Sulfates, amino acids also, because the mechanism, the proteins are same, but the mechanisms are similar. The transport maximum for amino acids is about so much, urate is so much, lactate is so much, even plasma proteins is so much. Okay, so we consider the limit within which the cells of proximal convolute tubule can reabsorb, and beyond the limit it is just not possible. Okay, are you okay so far? Hello, we are done. Good. Okay, this is very simple. We are just talking. We have done it already. We will do it once again. We have seen this image. The author makes a point in this slide that water reabsorption by osmosis coupled mainly with sodium. So if sodium goes, the water will go. We alluded to this point already. I can just move on. This is a box diagram, but we have done it, but let's do it once again. Sodium reabsorption. Why sodium go? Lumen negative potential, and then because sodium goes, then water goes, which the chloride go by the luminal concentration, the urea goes. This just tells us as to how are the different substances reabsorbed across the. One very interesting point. Please remember, bicarbonate ions are important for us. What for? pH. They play a very important role in maintaining. What am I talking about? Bicarbonate ions. So we want to conserve them. Okay. So here author tells us as to how are we going to reabsorb the bicarbonate ions. We don't want to lose them. Okay. So here we have the lumen, and here we have the apical surface, and on the apical surface we have an antipode system. And that antipode system does what? It's there. Exchanges, antipode exchanges. Sodium will of course come. How sodium will come, that we have already seen. Sodium come, and it will, and against that it will go the hydrogen ions will. Now on the basal side, of course we have the pump. Okay. Now here we have another protein system, okay, in which again it is an antipode system, which will, let us see, carbon dioxide, water abundantly there, carbonic anhydrous, H2CO3, carbonic anhydrase, carbon dioxide and water. Carbon dioxide and water absorbed in again carbonic anhydrase within the cell. Okay, this gives rise to H ions and the bicarbonate, and here something interesting happens whereas these H ions are exchanged for the sodium ions and H ions are being thrown out. Remember, two issues. We want to keep bicarbonates and we want to get rid of H ions. Why? Our body keeps on generating huge amount of H ions right from stomach, okay, to the metabolism of amino acids. We keep on generating H ions, and in the interest of pH, we better get rid of H ions and keep on conserving what? Bicarbonate ions. Okay. So there you have the, so here what we do is, here again we break up bicarbonate into H and CO3, whereas the H ions will go by help of this antipode system. We have already seen this, H ions will go there, whereas the bicarbonate ions, now there is another system of proteins on the basal side which will exchange, no, this is a sympod system where sodium and bicarbonate will be taken from the cell towards the blood side, and this by, okay. Can somebody please tell me what does this system just signify? It's exchanging what and what? Bicarbonate and chloride. That makes sense. Why? Because it is negative for negative. So you are bringing about the transport of substances without really making any change in the ion. So you are exchanging sodium for chloride. I'm sorry, not sodium here. Here the bicarbonate is exchanged for chloride. So what is the message in the slide? Please. These cells play an important role in the recovery of the bicarbonate ion. We don't want to lose the bicarbonate ion, and at the same time we are making sure that the H ions are got rid of.
Sir, in the last slide, carbonic anhydrase was shown on the lumen side, but since it's a protein, is it on the, say, luminal membrane of the?
It's there on the villi.
On the villi.
Everywhere. I'll just explain this slide and we'll close today's talk. What I'll do is, my proximal convoluted to be starting here and it's ending here. I'll take these 2 points, and I'll put them here, and from 0 to I'll divide it into 0 to 100. The distance 0 to 100. So 0 is right at the level of Bowman's capsule, and 100 is where the loop of Henle is beginning. Okay, number 1. Number 2, what I'll do is, I will collect the fluid, the filtrate, here at 5 different points, and those points I'll call as 20, 40, 60, 80, 100. So what I am doing, I am just collecting these samples, filtrate along the length of the proximal convoluted tubule from the beginning, 20% distance, 40, 60, 80 and 100. Are you with me so far? I am collecting that fluid, filtrate there, and I am subjecting it to, I'll take it to atomic absorption or whatever HPLC I have, and I am trying to subject it to analysis. Okay, and the concentration that I have here at 0 position, right, the fresh filtrate, what a dirty word, the filtrate, okay, is, I'll call it as whatever is the concentration, sodium, whatever is the concentration, I'll call it as 1. So whatever sodium I have here is, sodium is here is 1, okay, and I have sodium here evaluated here, and here is my last sample, and I find that my sodium is almost 1. Why is it still 1? Because lot of sodium is already absorbed. We have seen that huge amount of sodium has been absorbed. Why is it still 1? Water is also absorbed. Very simple. Water is also absorbed in the same proportion. Okay. As a result of that, the concentration of sodium ion has remained same. Okay. But if I see the amount of glucose here, and amount of glucose here, and I compare the amount of glucose here with the amount of glucose here, what picture do I get? 99% of glucose is absorbed. Are you with me? And same is also the story for yet another valuable molecule. What is the other valuable molecule? Okay. Are you getting it? Are you getting the profound importance of proximal convoluted? We have absorbed completely. By the time you are done with the proximal convoluted tubule, glucose is completely absorbed, amino acids are completely absorbed. At the other end, if you go to the top, you have another interesting story to tell. What is going up? Why is it going up? You don't want it back. You don't want. You see, exactly. The water has gone. Creatine has not gone up. Creatine, the creatinine per se has not gone up, okay, but because the water has been absorbed, therefore the concentration of creatine is relatively more, and therefore creatine will go. And we have already seen the importance of creatine. I am continuously making a mistake. It is what? Creatinine. What is it? Creatinine. And the creatinine, and then here is the osmolality is same. Okay, it's just one, and bicarbonate is here, sodium is here, etc. So this gives us an idea as to overall what is the rate at which the different substances are being absorbed as the filtrate moves along the proximal convoluted tubule from one end to another end. I'll stop now.